there. And so I'll just say a few things. So we made a few changes this year to increase the engagement with our students and ECRs. And so we decided to bring back the student / postdoc talk before our guest talk. That will be a 5-minute talk, and Daniel will keep you accountable for those 5 minutes. But that also means that the hub wings will now not be showed in these roundtables. And that is essentially because of time constraints. We couldn't possibly have it all. And so the hub wings will be in the newsletter. So please continue to send us all the amazing things that you're doing, papers, preprints, big study that you completed, milestones, progress reviews, and so forth. So let us know. We really want to celebrate all of your achievements. The other thing is after the talks, we'll have lunch catered as usual. And then after that, at 1 o'clock, we'll have a new thing, which is a Q&A session with the speaker. And that's just for students and postdocs. So lab-heads, you're not allowed. Don't go in. This is to allow the students to ask any questions. They can be scientific, they can be about career progression, development, anything. So take advantage of that. And I'm not sure whether we'll be here. Where's Sarah? Sarah will say, oh, where is the Q&A session? In here. It's still in here. So just stay on until 2, if you like. All right. Thanks so much. So Daniel will present the speakers.
Okay, so first we're going to have a student talk. So this is Jisun, who's a PhD student within the prediction and decision-making fund. He's going to be giving a brief talk titled Jointly Modelling Behaviours in EEG, an example with evidence accumulation models prior to our roundtable. So come on up.
Thank you for giving me this opportunity to share my work on joint modeling. So, yes. So I will first introduce a couple of terms what I mean by joint modeling and computational models. So computational models, I'm not used to this, can be generally defined as just mathematical models that explain behavior or neural data with some formalized cognitive and neural mechanisms. And in terms of joint modeling, what we're trying to do is simultaneously explaining behaviors and neural observations with either a mechanism from the behavioral models or linked to mechanism between the behavioral and neural models. So why we're doing this, some of the advantage will be simply those mathematical models itself are stronger tests or validation of existing theories, and it provides some mechanistic insights into the neural observations and signatures. And finally, by including more models, more data to be accounted by the model, we essentially including more constraints to the model as well. Today, I'm using some of my work with the recognition memory task, which is simple memory task where a participant first remember a list of items and they're later being cued to recognize those study items along with new items. So a ERP crawl list, which is named the late positive components, is consistently observed in this type of task. And as you can hear, it usually appears 400 to 800 milliseconds after seeing a probe for testing your memory. And it also consistently showed a higher amplitude for hits, which is correct recognition of study item, compared to correct rejection. And this component has been theorized to represent recollection, which is a retrieval of detailed memory during the recognition phase. And recently we have reviewed and summarized some of the features of LPC, and we found, first of all, LPC amplitude increased with better remembered items in terms of the memory strength. And it also increased with faster response times as well as higher degree of decision confidence. And importantly, what we found recently is that these LPC is actually peaked at the time of response instead of occurring at the fixed time window after participants see something. All of these features are quite related to a decision-making variable. That's why we're using the diffusion decision model. Many of those you may have known about this. So the this model essentially assumes the noisy accumulation process starting from a starting point Z and going through a noisy accumulation to either the boundary with the average speed of as accumulation represented by the parameter B. And while we're interested in the V parameter or the drift rate parameter is that similarly a higher drift rate will also be associated with a more accurate decision, faster response, and higher confidence. And that's why we're interested in joint modeling behaviors in LPC with some shared mechanism, which is the evidence accumulation. And what we did was to let DDM jointly predict behaviors in LPC with drift rate across trials. And a couple of things that we're interested in this from the research is how strongly these LPC changes as associated with drift rate and how this relationship unfolds over time. And because the really rather complex model, we train neural networks for likely free parameter estimation, which we'll not talk about here. So for the main findings, first of all, we have to make sure our model is accounting the data well. So the model can actually well predict across accuracy, reaction time, as well as LPC amplitudes. But what's interesting about this line of research is we can calculate the amount of variance in the LPC to be explained by trial variations in drift rate. And we found that the change in drift rate can be explained by drift rate, for change in LPC could be explained by drift rate, and also for target symbols. And this is consistent with the idea that LPC is more memory specific. And by using another model to predict simultaneously the changes in LPC across different time window, what we found is that this relationship in LPC and drift rate was getting stronger as the time gets closer to the time of response, which is consistent with that idea of the LPC peak at the time of response. So overall takeaway message, we have demonstrated LPC is likely to track the strength of memory across items during retrieval in recognition memory tasks. And this is consistent with one of the mnemonic accumulator hypotheses for the parietal lobe, where memory evidence is accumulated over time, which provided a sort of a potential mechanism account for the LPC function. And finally, we have demonstrated the benefit of the joint modeling framework, which can be easily applied to different settings and neuroimaging modalities. And yeah, that's it. Thank you.
Thanks very much. That's an admirable opening act. We're on to the main act. So our speaker today coming down from UNSW in Sydney is Professor Gavan McNally. Gavan is a professor of psychology at UNSW Sydney. He research in his laboratory focuses on the psychological and the brain mechanisms for associative learning and their roles in health and disease. He is senior editor at the Journal of Neuroscience. He actually handled one of my papers. Thank you, Gavan. Did it get it? And it did. And has received the D.G. Marcus Award from the American Psychological Association and the Pavlovian Award from the Pavlovian Society. He's a fellow of the Academy of Social Sciences in Australia, the American Psychological Association, and the Association of Psychological Science. Please join me in with Gavan.
Thank you for a very kind introduction. Thank you, Martin, for having me down, and thank you for coming today on what ought to be a great day. Certainly at UNSW, the campus is absolutely empty this week, even though it's the last week of our first trimester. So let's see. Which button was that?
Probably before.
And it should work now.
There we go. So I will thank everyone again at the end, but everything I'm going to show you today is a product of work by a lot of people, including Liz, who was here as an undergraduate student, and I'll show you Liz's data. Liz came up to do a summer scholarship with us at the end of her third year before she did honours in this building. And that was fantastic. But these are the people in the first three rows who did all the work, and the people in the bottom row family and literally, and also collaborators without whom none of this work could happen. Reviewer 3 said this to me, and they weren't, I think, talking about the decision to submit the manuscript. At least I hope they weren't. Their point was that poor choices are part and parcel of being. We all make some bad choices some of the time. We might buy things that we don't need. We might speed or engage in other risks that at the time seem like they're worth it, but they may not be. And should we, as it turns out in the unlikely event, experience some negative consequence from those poor choices, we learn from those mistakes and we hopefully make a better decision in the future. In associative learning, we call this punishment learning, right? And it's learning about the relationship between your actions and their consequences, and specifically their adverse consequences. And it's a form of instrumental learning, and it's a form of learning that allows us to avoid harm. But some people, as we know, make persistently poor decisions. This is my gym, and this is my preferred treadmill where I try and forestall and prevent the ravages of aging. But in order to get to my gym, I have to walk through a room full of poking machines. And this is 500 meters from my home. And it's just one example of the kinds of persistent poor decisions that people can make. And so we are a very small country population-wise, but as many of you know, we have 20% of the world's pokie machines outside of casinos. 1 in 5 are in Australia. And the average loss per person per year off a pokie machine in Australia is $1,300. But those averages aren't meaningful, because I'll probably bet that none of you lose $1,300 a year on the pokies. They're isolated to a much smaller number of people with problem gambling. And that's just one example, I think, of a variety of behaviours that people persist in that have adverse consequences. And the argument is, right, that these people appear to have some difficulty with learning about those adverse consequences. And this idea is embedded into diagnoses of those disorders. And it captures the frustrating to the external person, to the family, to their communities, the frustrating but important point that people are continuing to engage in behaviours that are causing suffering to themselves and to others. And we're interested in why. And one of the ways in which you can try and answer that is to think about the brain mechanisms of learning about the consequences of our action, as KDATK actually asked on social media. And the answer that's normally given to that question is that dopamine is somehow important for this learning. And specifically that rewards will promote actions because they lead to increases in dopamine, and that dopamine increases a teaching signal that reinforces the action. And punishes will cause a reduction in dopamine. That reduction in release of dopamine is also a teaching signal that decreases the probability of the action that gave rise to the consequence that led to the dip in dopamine. That's an interesting idea. It's effectively the simple summary of reinforcement learning. But it's not a new idea at all. It's one of the oldest ideas in experimental psychology, because it's just a rewording of Thorndike's law of effect from the 19th century. And even Thorndike 5 years later didn't believe this account of punishment. He retracted it. He said it can't possibly be the case. Even though he ended up retracting that view of punishment, it's persisting into modern neuroscience and into modern reinforcement learning. And what I want to persuade you of today is that learning about the adverse consequences of our actions is not just, and perhaps not even, about dopamine. It's far more interesting than that. And more specifically, if we want to understand why people persist in behaviours that are having adverse consequences, then we need to understand how they learn about punishment, and we need to understand what they learn about punishment or about their actions. And we do this, much to my surprise, across 3 species. We work in humans, we work in rats, and we work in mice. And today I'm just going to show you data from my 2 favorite species, which are mice and people. I used to love rats, but you'll see why. You can do so much in mice these days. It's crazy. So the poor old rats are being phased out. So the first part is in mice, and it's asking the question about brain mechanisms of punishment. How do we learn about punishment? And this work has been led by Philip John Richard Depressel, Alexander Gregory, James Diefenbach, Jess Chen, Lucy Choi, Nikki Huang, Zara Nguyen, and Vishwan Lin. And we started this by taking full advantage of things you can do in mice that you can't do in other species. And one of the things that you can do in mice is you can generate whole brain maps at single cell resolution of cells that were active in a recent period of time. Okay. And that's what we did. So we have mice and they're in a task. They get 10 days of lever pressing, and there's 40-minute sessions. The lever comes out. If they press it, they get food. Goes back in after 5 minutes, another lever comes out. If they press it, they get food. They get 10 days of that, so they're responding well on both levers. And then we introduce on one of those levers only a punishment contingency, so that the 10th response, for example, on one of those levers also earns a mild foot shock. The other lever just continues to earn food. We have in this experiment control animals who never receive shock, so they just receive the food rewards. And the obvious thing to do if you're a mouse, a rat, or a person is just to withhold responding on the lever that's causing the shock. You only have to wait 5 minutes till the other lever comes out, and you can knock yourself out earning pellets off that lever. Right? It goes back in for 5 minutes, the punish lever comes out, just withhold responding. You're not going to get shocked if you withhold responding. And that's exactly what our mice learn to do. This is across the 4 5-minute blocks. And it's a ratio of their lever pressing. As that ratio goes down, it means that they're selectively suppressing the action that's earning punishment. The controls continue on equally, responding equally on both levers, right? So they're learning to reduce the behavior that's causing shock. Then we take their brains out, and instead of cutting them up, we clear them. So we improve the refractive index of the tissue so that we can actually image through the entire structure without having to section it. And before that, we clear the tissue, we perform whole brain immunohistochemistry to label for nuclear proteins that indicate that the cell was active during our session. And then we image the whole brain, right? The whole intact brain in a light sheet microscope. And we count every cell that's stained, every nucleus that's stained with the activity marker, and we align that to a standardized map, the Allen Institute map of the mouse brain. And if you do that, you can study the mouse brain at various levels. It's what's called level 6, which is dividing the mouse brain into 206 different brain regions. And we're effectively plotting here functional connectivity, which is the correlations between the counts of our nuclear activity proteins in different brain regions, from the very, very front of the cortex to the very, very back of the brain stem, across our punishment group and across our control group who've just received the reward. You can do a lot of things with those data, but one of the things that we do, which always better to pick what other people are doing sometimes and create your own thing. So of course, in cognitive neuroscience, in whole brain imaging, it's very common to use network and graph theory to analyze data. So that's exactly what we've done. And what we found is that both the punishment whole brain network and the reward or the control whole brain network are similarly modular. There are 17 modules or communities in the punishment brain. There are 16 in the reward brain. And they have, as one might expect, for those people who study brain networks, they have other properties, one of which is that they're scale free. And what that means is that if you line up all of those brain regions and you threshold that matrix to only include correlations between regions, say greater than 0.8. And then you look at the number of functional connections between greater than 0.8 between each brain region, and then you just line them up in terms of how many brain regions have this many sets of connections. You find the same shape distribution, which is that most regions have relatively few functional connections, but some regions have many. And that's true across both the punishment network and across the control network. So structurally, those whole brain networks are very, very similar. But the components of those networks are quite different. So the regions that have the highest functional connectivity with other regions are shown in red for the punishment network, and for the control network is shown in blue. And you don't need to understand mouse neuroanatomy or neuroanatomy at all to be able to recognize that those two maps of whole brain activity are quite different. What do you do with that, right? If you're in cognitive neuroscience, then you just run some Python script and you attack that network, right? You can delete nodes in that network. There's however many nodes in each network, and you can delete the top 10% of nodes to ask what effect does that have on network structure, where your metric may be the average length of the shortest path across the network, right? So like the train network, you might take out a station to see what that does to the movement of the trains. You might perform that deletion randomly. You might target the top 10%. You can do, well, we did all that. You can do all that as well. But because we're working in mice, we can actually do the experiment where we silence key players in that network. And we use a chemogenetic tool. These are tools, viral vectors that allow us to express G protein coupled receptors into neurons, and those G protein coupled receptors don't recognize anything produced by the body. So they don't bind any neurotransmitter, neuropeptide, etc. But they do bind a synthetic drug that we can then inject into the mouse. And those receptors in this case are inhibitory. So when we inject our drug into the mouse, we can silence the cells that are expressing our tool, and we won't have any other effect on the brain at all. It's quite a powerful tool. So that's what we did. But which regions to silence? We looked at all of the brain regions, and we looked at those that showed the greatest change during punishment learning, the greatest increase in functional connectivity. And number one on the list was, I hadn't heard of this before, which is the rostral linear nucleus of the raphe. And the joke I like to say about this, I learned neuroanatomy from my PhD co-supervisor, who was George Papsanos, who made atlases of mouse brains and rat brains. And George taught me that as a shortcut to study for exams, he said, whenever you see raphe, just think serotonin. Of course, most serotonergic nuclei are in raphe. But the rostral linear nucleus of the raphe doesn't contain cells that make serotonin. It contains cells that make dopamine and glutamate. And it's located, if you're interested, in the midbrain, and it's located just above dopamine neurons in the midbrain. The zona incerta is located behind the striatum, just next to the amygdala. And another region that came out is the basolateral amygdala, which we were grateful to see. So we expressed our tool, our inhibitory dread, in those three regions, right? We expressed it bilaterally in the basolateral amygdala, bilaterally in the zona incerta, and bilaterally in the rostral linear raphe nucleus. So now we've got these three brain regions expressing our tool, and when we inject our drug, we can selectively silence those regions to ask what does it do to punishment learning. The ligand that we use to silence those cells is a metabolite of clozapine N oxide. So we did that. We silenced those regions prior to a punishment learning session. And what we found was that the silencing of those regions greatly impaired punishment learning during the punishment session. relative to controls, and that was selective to injection of our ligand, because if you inject a vehicle, you don't impair punishment learning, and that impairment in punishment learning carried over the next day when the animals were tested drug-free, CNO-free. Okay. So what happens at the cellular level in these regions as animals learn about the relationships between their actions and their adverse consequences? To do that, again, this is why we're using mice. We have a very cool set of tools, but one of the coolest ones are these miniature fluorescent microscopes that weigh about 3 grams, and you mount them on the head of the animal, and they allow you to visualize single cell activity. And the way that we do that is that we use a viral tool to express a genetically encoded calcium sensor into cells. This sensor shows increases in fluorescence as calcium levels inside the cell increase. As calcium levels inside the cell decrease, the fluorescence signal comes back down. And in order to see that fluorescence signal, you have to provide an input light to drive the fluorescence signal, and you have to be able to capture the emission of that. And that's what this miniature microscope does. And in order to image deep in the brain, we have to put in a lens. So we put in the gradient refractive index lens into or above the basolateral amygdala. And what that looks like is something like that. So it's not in real time, it's a bit faster. There's one of our critters going about her world, responding on levers on this side to pick up food on this side. And I'm not sure if you can see, but each of these is a cell in the basolateral amygdala. And we image from these cells throughout the entire session. We imaged over 1,000 cells from our 6 mice in this experiment. It does take a while to do, but not as long as the analyses. And those animals were trained in the task I just described, so they learned to leave a press for food, and then we punish one of those behaviours but not another. The animals suppress the punished behaviour relative to the other, and then we can extinguish that punishment by just removing shock, and the animals quickly adjust their behaviour backwards and back to normal. And the question is, what's happening in the amygdala? Well, there's a lot of things, but one of the things that happens is that if you track only the cells that you can guarantee are the same across each of these 4 periods of the experiment, we can then cluster those cells, and the clustering identifies 3 clusters. The responses of those 3 clusters to punishment and reward are shown there. Cluster 1, which is a light blue there, teal, shows generally inhibitory responses to punishment, generally inhibitory to reward. Cluster 2 neurons show excellent excitatory responses to the punisher itself, but they're showing inhibitory responses to the reward. And cluster 3 neurons are showing excitatory responses to both. Okay. So there's quite a bit happening, even in just one of these regions in this task. But what's even more interesting to us is how those clusters relate to the behaviours, the actions. And the top row are the activity profiles of each of those clusters as the animal zeroed around the action. A lever press, punished lever press on the top, unpunished lever press on the bottom. Those coloured lines underneath, we created a bootstrapping procedure for this approach. So they represent bootstrap 95% confidence intervals when those for the periods of time when those waveforms do not include 0 in them. So they're significantly different from 0, if you like. And what you can see is that generally the response of the 3 clusters to the control lever, to the unpunished lever, is not changing during reward, punishment, early punishment training, late punishment training, during a choice test, during extinction, and then late extinction. But to the punished lever, there are changes. And specifically, cluster 2 neurons show this emergence of excitatory transients as prior to and as the animal makes that response that risks punishment. And that's contingency specific because it's lost or reversed during extinction. A different way of visualizing that is just a fit-based way. And cluster one, and that gives you a better idea of what those clusters might be doing. And cluster one seems to differentiate reward from everything else. Okay? Cluster one treats a punished lever press and unpunished lever press, the punisher itself, as the same. It just treats those as different to reward, to the ingestion of a food pellet. Cluster two neurons look complicated, but they're really not. They're quite simple. Where what they're doing is initially, if you just look at the top quadrant, initially during punishment, the punished lever, activity around the punished lever is very similar to reward and very dissimilar to punishment. But as the animal learns punishment, the activity around that lever press becomes similar to punishment and very different to reward. You can put that into like MDS, you put that into a 2D space, and that shows very clearly that the punished lever press becomes very similar to shock punishment during punishment, and as you remove the punisher, it moves back to this other space where it's now similar to reward and to other events. And the third group of cells, or the third cluster, is especially interesting to me because it seems to partition out antecedents and consequences. That is to say, it treats the actions as similar and different to the reward and punisher, but it treats the rewards and punishers as relatively similar to each other. Okay? I'll provide a pithy summary of that in a second. So what are we doing with this now? The next step is cool. I don't know if this is going to tell us anything new, but I had to put it in because James in particular has been working very hard on that. We're using machine learning based full capture of the animal's behaviour during this task. We're identifying specific behavioural syllables that might only be a couple of hundreds of milliseconds long. And we can then compute, as an example, transition probabilities between each of those syllables to understand how behaviour is being constructed in this task, right? Beyond just this measure of lever pressing, what is the animal actually doing? And then how is it that the introduction of reward or the introduction of punishment changes the structure, this microstructure of the behaviour? And this can occur across minutes within a session, but you can also track those transitions across days or even longer. So we think punishment's far more interesting than dopamine. Firstly, we know that it's associated with a disengagement of regions that are linked to motor performance, frontal cortex in particular, and the engagement of a large network that includes regions important for emotional learning, like the basolateral amygdala, reward and aversion, the nucleus accumbens, stress, the dorsal raphe, etc. And we can go into, and we are, and I've just shown you one of those regions, which is the basolateral amygdala. We can go in there and we can image the activity of cells, of neurons at single cell level and hold that cell for at least 10 days across our imaging protocol to ask how single cell activity changes across learning. And in the basolateral amygdala, what you find is that that activity parses what we think of as a minimally sufficient syntax for learning. One cluster of cells seems to especially like reward. A second cluster of cells seems to partition the antecedents of an event from the event itself, actions from outcome, responses from consequences. That was cluster 3. And a third cluster, which is cluster 2, shows a punishment response specific transformation in its encoding of that action. And it treats that punished behavior as similar to the punisher itself. Okay? I've given this talk a few times. No one ever asked me anything about all this stuff. They always ask me about what comes next. So that's nice. we're using mice because we can do a bunch of things in mice that are just impossible to do in rats and people, right? In terms of getting an understanding of biology in this process. But we also study people because you can do things in people you just simply can't do in rats and mice. So I want you to imagine that you're a space trader and your goal is to accumulate points, and you accumulate points by trading with 2 planets. Yeah. Okay. You initiate a trade by clicking on the planet, and 50% of the time will give you 100 points for that. What do? Well, you should learn to click on the planets and accumulate points and watch your points grow in a predictable fashion. Then in a second phase, we make things a little bit difficult for you. We arranged that some probabilistically responses on planet 1 will lead to a ship appearing. This is a pirate ship. It's bad news for you because when this pirate ship appears, it will steal your points. How many points it steals depends on the experiment, but it will take your points. When you click on planet 2, with equal probability, another ship appears, but this ship is harmless. It doesn't take your points at all. And the question is, what do? Well, you should learn reasonably quickly to reallocate your behaviour away from planet 1 and just start responding exclusively on planet 2. They're both presented concurrently. One never goes away. You can just keep clicking planet 2, accumulate lots of points, because clicking on planet 1 earns you punishment. This is a conditioned punishment design. The punisher is the ship followed by points lost. And we've done this across almost 2,000 people now, but this was the first time we did it. And what we found is that some people were very good at learning to avoid punishment. So they would suppress their responding on planet 1 relative to planet 2. But some people did not. And that's a clustering analysis based on the performance at the end of the experiment. Okay, so there's some people who learn, they're sensitive, and there are some people, even in that simple task that I just showed you, who don't learn. They're insensitive. They don't learn punishment, is what I should say. We didn't actually start with this task. We started a couple of years earlier with this task in rats, so there is some rat data here. This is exactly the same logic, except that we have rats responding on levers for food rewards, and they're being punished by presentations not of ships, but of foam followed by shock, and not punished when a light comes on. And you see exactly the same thing. These rats, some of them learn to reallocate their behaviour. So they learn to avoid punishment by suppressing the punished action. Some of them do not. Some are sensitive, some are insensitive. In people, that insensitivity to punishment is occurring despite them being highly engaged in the task. So you always get, are they engaged? Yes, they're engaged. This is the number of clicks per minute that the participants are making, the number of trades per minute that they're initiating with the planet. So they're making 40 odd responses a minute across the insensitives, across both responses. The sensitives are making 50 responses a minute on the planet that's earning reward, and they're almost completely suppressing their responding on the behaviour that's earning punishment. So they're showing excellent discrimination. But both groups are clicking. And we know exactly where they click because we track the XY location of their clicks as well. The consequence of that pattern of behaviour is that the sensitives accumulate a lot of points that in most of our experiments are exchanged for money at the end. And the insensitives, they don't accumulate very many points at all because they keep making the same mistake. They keep clicking on the planet that's earning the ship that takes their points. They can't seem to change, as it were. Everybody likes reward. Nobody likes punishment. We ask them to rate the value of reward, and we ask them to rate the value of punishment, and they tell us, yep, I like earning points and I don't like losing points, right? Everybody knows which ship is bad news. They can tell us that, yeah, this ship leads to attack, this ship does not lead to attack, and our sensitive and insensitive participants are fine at that. And the consequence of that is that they place different values on those ships. So they tend to value or like the ship that doesn't lead to attack, and they dislike the ship that does lead to attack. Where they differ is in the values that they're placing on those actions derived from the knowledge that they have about those actions, which is to say, look, the sensitive participant know that when they click on planet 1, response 1 leads to punishment or leads to ship, response 2 leads to the other ship. The insensitive participants don't learn that. I'll show you what they learn. And the consequence of that is that the sensitive participants value the action that's not punished, and they don't value the action that is punished, whereas the insensitive participants equally value both actions. And what's happening here, and the reason why we're using people, is that we can probe what they learn in this relatively simple task by getting them to rate likelihoods of each possible set of combinations. How likely response 1 is to lead to the shift 1, how likely shift 1 is to lead to attack, how likely response 1 is to lead to shift 2 to attack, response 1 to shift 2, etc. All of it. And what we can show is that the sensitive participants learn this task easily and quickly. They learn response 1 leads to shift 1, which leads to attack. Response 2 leads to shift 2, which has no attack. The insensitive participants, they're not stupid. They just don't learn the same thing. They are learning. But what they're doing is variable across them. But generally what they do is they mix up how their actions are causing the shifts to appear. They tend to be, as I showed you before, very good at learning this ship is bad news and this ship isn't. What they struggle with is correctly identifying how they're causing those ships to appear. But the cool part about all of that is that regardless of if your model is correct or incorrect, it's coherent. What I mean by that is that your rating of how likely response 1 is to attack is predicted by your ratings of each of the individual components of your model linked to response 1. Yeah, which, if it wasn't like that, you'd be worried because it should be coherent. But that just shows you that our insensitive participants have a coherent but incorrect model of how their behaviours causing adverse consequences. So that should be easy to fix, right? Incorrect learning, man, I learned a lot of incorrect things. So it shouldn't be a problem. We should be able to correct their models because we learn in many ways. We learn from experience, we learn from observation, we learn from instruction. We're very good at integrating knowledge from different sources. This is precisely why we use things like warnings in public health. So we gave a cognitive intervention of sorts, is where we had participants play this game again, 3 blocks, just like I showed you. And then we told them exactly what was happening in the task. And this is exactly what came up on the screen. Local intel has determined where the pirates are coming from. Click next to view this intel. And then we lay it out for them precisely as it is, right? And then they have to pass a knowledge test. They have to on-screen knowledge test before they can play the game again. And that's the test, okay? And that information turns out to be quite effective for some people and completely ineffective for other people. What I mean by that is that we run them through the task, we provide them with the contingency knowledge, and then they can play the game again in one more block. We perform clustering last on a two-step clustering across here and here. And you see prior to contingency reveal exactly the same pattern that I showed you before. Some people learn from experience how to avoid punishment, so they suppress their behaviour. Some people don't. They're still losing points. They're making the punished response when they shouldn't be. And then we tell them what happens. And that splits these insensitive people into two kinds. Some who respond to that contingency information immediately by changing their behaviour, right? They got it wrong. We tell them what the correct way of behaving is, and they switch completely. And some who do not. And so the ones who change, we call unaware, because they were unaware of the correct structure of the task, but when you tell them, they change. And the ones who don't change, we call compulsive. It doesn't matter if you're sensitive, if you're unaware, or if you're compulsive. People know what they're doing. They know why they're doing it, and they think what they're doing is the best way of behaving. This is the behaviour. This is a response to questions, post-block questions. How did you allocate your behaviour in the previous block? So it's the one possibility, right, is that people just don't know what they're doing. Their behaviours become so automatic in this task, in theory, it could be argued that their behaviours become automatic in this task. They don't really know where they're clicking. They're just clicking. That's not the case, because when you ask them how did they allocate their behaviour, you're asking them after each block, it looks exactly like what they did. So they know what they're doing with great accuracy. Even more interestingly is when you ask them after each block, what should you have done to maximise points? This is what they tell you they should have done. So what they should have done is what they thought they did, which is what they actually did. And again, this has to be the case amongst most people, you think, right? Behaviour should align, they should have knowledge of what they've done, and the behaviour should align with their task goals. But what it shows you is that even in this simple task, people form three different understandings of how to behave optimally. But for each of those understandings, behaviour is deliberate and it's aligning with their task goals. Okay? Why? Well, part of it has to do with the very fortunate fact for us as a species is that generally we're pretty lucky. Negative consequences from our actions are rare, generally. And in most cases, they're probabilistic, right? If you speed, you've got to be pretty unlucky to be caught. Yeah. And you're certainly not going to be caught every time you speed. So what role do those experience contingencies play in people's capacity to learn about, through experience, about their actions, or to learn from instruction about those? So we had 3-group experiment, exactly the same as I just showed you, except that one group is being punished at 10%, one at 20%, and one at 40%. And there's a complexity in here because we match the punishment value across those. So if you were punished at 10%, then you lost 40% of your points when you were punished. And if you were punished at 40% of the time, then you lost 10% of your points each time, etc. But what happens? The same. In this case, we have the same 3 classes of people from the task. These are different data, different people to what I showed you before, but it replicates every time. But more interestingly, the contingencies that the participants were in dictated the phenotypes, the probabilities of the phenotypes. And the important point is this: when punishment is frequent, 40% of the time when those individuals click on that planet and they get punished, most of those participants either learn from experience that that's going to lead to punishment, or they respond to instruction, right? If they haven't learned from experience, they respond to instruction, so they're unaware. Only 12% of those participants were classified as compulsive when punishment was frequent. But when punishment was rare, 45% of those participants were classified as compulsive. So it's interacting with the frequency of punishment and rare punishment. My colleague Brett Hayes sent me down a rabbit hole of learning traps. I don't know if anyone's read any stuff on learning traps. It's fantastic literature. But I like the idea of learning traps, and rare punishment acts as some kind of trap, right? Because at low probability, these individuals are more likely to be compulsive, but at high probability, they seem to be protective. Yeah. Okay. Are they stable? This is what Liz did. Liz came up and she had 6 weeks in the lab. And so we wanted to run an experiment. And in this case, we ran this one on prolific. And Liz recruited a lot of people, but I'm going to show you 128. And she had them play the game twice. She had them play them over the summer of 2023. And then she came back down here, started on this, and then halfway through the year, she recollected data from them 6 months later. Okay? And this is what they look like in the first test. And playing the same task, and again, you get exactly the same 3 phenotypes. Some people who learn from experience to avoid behaviours that have adverse consequences, some who do not. And of those who do not, some respond to information intervention and some do not. Okay? And then she had them play it again 6 months later and found pretty much the same profile of data. But of course, the really interesting question isn't just demonstrating that you can get the same pattern of data 6 months later, but are they the same individuals across those 2 time points? And so we learned how to make a Sankey chart. And the short answer is yes, they are by and large the same individuals across that time. Most people are identified at time 2 as belonging to the same cluster as time 1. And at this point, we did counterbalance all the different responses and all the different shifts. We also asked them if they remembered what had happened. They, as unsurprisingly, they couldn't remember what happened 6 months ago. So we don't think this is simply some carryover effect from the task itself. We think that the task is tapping into something that's a little bit more reliable, identifying the different ways in which people learn about the adverse consequences of their actions and respond to interventions about that. There's 2 things about this slide that always amaze me. The first is that I'm actually putting up a slide that's got all those acronyms on it because I don't work in that space. But the second is that, look, we've measured a lot of people now, and we've also on that task, and we've also measured them on a lot of other things. Many, like all those things for starters. None of it matters. The 3 phenotypes don't differ on any of those measures. Maybe that's because those self-report measures occupy a different space to behaviour. That's an interesting idea, but they're not helping us predict or parse out the data. We've done a lot more experiments than I've shown you. For example, we've shown that even providing that information early, providing the explicit contingency information before the participants start, telling them exactly what's going to happen, that doesn't help. You see exactly the same split across 10%, 40% contingencies if you give that information early as if you give that information late. Inspired by work, some of it from here, about the impact of warnings and inoculations, we've even tried those and they don't seem to help. Why do these differences matter? Well, most, if not all, problem behaviours are accompanied by a lack of recognition, correct recognition that those behaviours are causing harm. And certainly in the space, one of the spaces work with Giselle a couple of years now already, one of the spaces where that's absolutely true is in alcohol and other drug use. And lack of recognition that your alcohol use or other drug use is causing you a problem is the number one barrier to seeking treatment. Stigma is number two. How can you not know that your behaviour is having adverse consequences? Well, Hannah Picard is a philosopher at Hopkins. Serge is a neuroscientist at Bordeaux. They have a couple of fantastic papers, but I'm not sure if any of you have ever heard of Hannah Picard or read her work, but if you're interested in psychopathology, her work is very interesting. She's argued that people don't correctly recognise the adverse consequences of their actions because they're in denial. And she means by that a form of motivated belief or self-perception where the costs of the behaviour are hidden to the individual. Okay? There's a corollary of that I like. And the corollary of that is that the cost of someone's alcohol use, for example, could well be hidden to them. But the benefits of their use, of that use, could be hidden to us as an external observer. Yeah. So they do not know that the choice to act is the cause of negative consequences. So she writes, there's no puzzle as to why drug use persists despite negative consequences if those consequences aren't straightforwardly known. And you think, how could someone possibly not know? And then you think back to what might have smoking, for example, in the 30s, 1930s, 1940s, 1950s, and you realise, well, there's probably good reasons people don't know that behaviour is having these adverse consequences. So I quite like that idea. I just don't know how to conceive of it psychologically beyond something like a form of cognitive bias or motivated belief. We have a different idea. And the idea is that in some individuals, there's a disintegration of the normal integration that you see between cognition, affect, and behaviour. And as an example, what I want to show you is, this take forever to make, that's why I wanted to show it to you, and it's useful. This is, obviously it's a 3D graph, and what it's showing you are how individuals value punished actions and unpunished actions, their punishment contingency knowledge, how likely an action is to R1 in our task, R1 leads to punishment, R2 doesn't, and their actual behavioural allocation, R1 and R2. And you want to be living down here. That's where you want to be living, because that's where you're telling us you don't like the punished action, that's 0. You know that that behaviour is leading to punishment, and you're withholding that behaviour. So you want to, your trajectory wants to be heading from the middle of this cube down to this bottom corner. And that is absolutely the case for people who are sensitive to punishment, and it's absolutely the case for people who are what we call unaware, and it's absolutely not the case for people who we classify as compulsive. And when you break that down, what you see specifically is that all of the groups can estimate their behaviour, as I showed you, and that relates very well to what they actually do. All of them can endorse their action selection, and that relates to what they do. But where those phenotypes are differing is how their action valuation and how their instrumental punishment knowledge is relating to their behaviour. It's a breakdown somewhere between their punishment knowledge valuations and then what they think is the best thing to do and what they actually do. Whereas the other two phenotypes are very similar there. So animals, what's learned during punishment? When we started this, I thought it would be like response, outcome, done. Animals, including people, form models, mental models of the consequences of their actions, and they're using these to guide their behaviour. That's relatively obvious. It's differences between individuals in the content of those models, despite the same experiences, that we think drive these robust differences in punishment-related choice. individuals may persistently make poor decisions because they overvalue reward over punishment, of course. I just might value or like the reward more than I dislike punishment. And so I'm going to choose that thing that I like. I might even make a poor decision because my choice is automatic. It's become habitual. We don't have a problem with either of those two options. But what we want to say is that people can also make poor decisions because of differences in what they learn from their actions, despite those same experiences, and integrating their actions with their knowledge, their instrumental knowledge. And a key factor that drives emergence of a problematic phenotype is rare or infrequent punishment. So for some people, achieving correct recognition of the adverse consequences of their actions is difficult. And even if they achieve that recognition, it may only weakly affect their behaviour. And with that, I would end and re-thank everyone in the lab and other labs who helped us with this. Thank you.
All right, we have a little bit of time for questions, and we'd like to encourage students to ask questions first. Yeah.
Thanks for the great talk. Now, I would like to just ask the same question. Did you check the early and late effect in each block in both group, especially in the in the group, like early effect, they are 40%. How was the performance in the first initially and then later?
Well, how does this look across that? How does a graph like this look across the contingencies?
It's represent one block now, right? One,
Yeah.
3.
Yeah.
And I want to ask,
Within?
did you check within the block, the start and the end? There is a possibility in the start they give a response, but later they just give up and they just adopt the.
Yeah, so we have response rates within each block and then consistent across the blocks. So the engagement, the clicking is consistent, and the data within a block almost always looks exactly like the average. There's no real changes across up or down within the 5-minute block. Yeah. Good question.
Any other student questions before we open the floor?
Thank you. That's a great talk. Sorry, this might be a bit tough, naive question, but the compulsive group, when they explain the pattern that when they explain what did they do, did they explain it in a pattern, or is it they explain it just randomly?
So what we get from we've done open-ended responses. We've obtained open-ended responses from hundreds of these people, and we've tried to pull something out of that. We've been unable to. But we're interested in collaborating with colleagues who specialise, and that's a pretty special skill, being able to extract useful information out of pages of self-reports. But what we do get them to do after, and this is why this class is not always a lot of fun, we do get them to rate probabilities of the different events, every combination of events after each block, and that's where we're getting their understand what we take to be the models that they're forming the task. And the problem is they don't correctly learn how clicking there causes this shit to appear and how clicking there causes that shit to appear, which is remarkable that they do learn. They just think that clicking here sometimes causes, clicking there sometimes causes this shit, but it also causes this shit, and clicking there sometimes causes this shit and also causes that one. That's what we take to be the core deficit difference.
Thank you very much. Very interesting. I just have a question about the first part of the talk. You presented the task where for punishment you always need to withhold the response, and for reward you continue. If you were to switch that, for punishment you needed to press, and for reward you need to withhold. Do you expect the components to change, or the clusters to change?
Maybe. So that would be something like active avoidance, right? So if you have to act to avoid something negative, that would be avoidance learning. Punishment is passive avoidance learning. Avoidance learning is active avoidance learning. Possibly, it would be an interesting experiment. What we know about the circuits of the brain mechanism thus far of active avoidance learning is that we're pretty confident that they overlap in the amygdala, in the nucleus accumbens, in that list of regions, and then there's dozens more. The one I think that'd be interesting would be the sensory motor regions, because if you have to make a response in order to avoid it, maybe those ones are going to pop up. Active avoidance learning is very complicated. I'm not sure if you've ever studied it before, but historically, it's been a fracture point for theoreticians in trying to understand what actually reinforces the action there. So in punishment, it's clear. The punisher is suppressing the action. In active avoidance learning, it can be less clear what an animal, including a person, learns about an event that doesn't actually happen. So that's that kind of difficulty there too. Yeah, but it's an interesting problem.
For the human data, I was wondering what the proportion of people are sensitive, because it seemed to depend on the frequency of the reward.
Frequency of punishment.
Punishment,
Yeah.
right. But then it looked quite low in like many cases, and I wonder whether it seems actually this typical thing is not to actually be not to be sensitive.
So if the stronger we make the punishment, the more likely it is to be sensitive. So if we go from 40% to 70% on the second sensitive, the task is, as described, it seems simple, but if you're not given any of that information, right? If you're just sitting down and told accumulate points, and then, well, be careful, space traders are coming and space pirates are coming and they might steal your points, then it can be difficult, especially at 10%. I mean, that's very weak contingency. So I wasn't surprised at all that relatively few people were sensitive at a 10% contingency when they're making 40 responses a minute, right? And 10% of those if burning ships. So we can pull that all the way around and just go up, yeah, which is kind of the point. And there's some sweet spot around here where we can still preserve all three. But it's yeah.
I was just wondering whether it's actually the sensitive people who are a bit maybe special in being really good at tracking.
Could be. Yeah.
And that's the norm is actually not
Yeah.
to be.
Yeah, they could be. And the ones that we actually like are the unawares, because they're the ones who respond to intervention. They're the ones who haven't figured it out. Only punishes they don't like. They don't know how to avoid them, but when you tell them this is the way to go, then they can avoid them. Yeah, it's a good question.
Yeah, I had another question, but this is too interesting. So maybe this plot here in these experiments, I wonder. So a lot of this is through the lens of sort of punishment as relevant to these sort of applications. But have you got any data, or have you thought about sort of how much this might also reflect sort of basic contingency learning? It reminds me in one experiment we ran, we told them probabilities of different things that are not associated with rewards, and there was still this subset of people who didn't really act like they knew that at all. Do you think there might be some association, especially in sort of those people who can't seem to learn that, that's not specific to punishments, but might be related to a sort of deeper inability
I hope so.
things?
Yeah, I hope so. And actually the literature, well, one of the literatures getting to the description experience literature, because it's very similar in the sense of differences in decision making from the description, which is abstraction, versus from experience. It kind of looks a lot like that. But the other one is, so ones we get asked a lot is, well, that doesn't have to be punishment. What happens if that's a jackpot? What happens if that's more reward, right? So is it as it were valence or not? Right? Is it their difficulty with that, as you suggest, with the task involved, the various processes, cognitive process underpinning performance, or is it selective for punishment? Is the question. And we haven't done the reward one yet, but in theory, we know. Well, we've done it in rats. We know in rats that task works. If instead of adding as an outcome punishment, you add in as an extra reward. That's CSC, which is an extra reward. Then they should do more of that. Yeah, so it'll be interesting to see people.
Two very quick questions. Firstly, you said just briefly in response to the question that this you can continue to drive slower. So if you make punishment contingency, close to 100%, does the compulsive group drop away entirely?
Yeah, you lose them, right?
Yeah.
The stronger the contingency.
And so you can get it down to near zero.
Yeah.
The second question is more like an exploratory one, like to what extent could this behavior be adaptive in other circumstances? Like, are they under some kind of counterfactual reasoning? Are they trying to figure something out that isn't clear?
Yeah.
Like, could the compulsive behavior in a population of individuals,
Yeah.
for a society, for instance, like under some circumstances, is there a set of circumstances where the compulsive, you want the compulsives in the room?
Yeah, maybe getting the PhD.
Yeah, exactly. And so I'm kind of curious, like, is there a way in which you can make explain the compulsive?
I think so. I think there's it seems relatively robust when we've done, and it's highly replicable and is persistent within an individual. So is consistent, I should say, within an individual. And you could think of it, if it wasn't punishment, if it were reward that you're getting at the end of that rather than punishment, then that would be a fantastic way of performing, right? It's that the consequences are adverse that make the behaviour problematic in this setting if the goal is to get the accumulate point. So yeah, I think so. I mean, I think in another task you might label that as resilience, except the problem is that the model that they've got is wrong.
Yeah.
So it's incorrect, and it resists both experience and description driven updating. And so yeah, you can imagine there are instances where that would be useful. They'd still be risky, I would imagine. Yeah.
Yeah, like they don't trust the experiment.
Yeah.
Like you're going to change the contingency.
Yeah.
Or something
Yeah.
like that.
Yeah.
See a lot of this in placebo, open label placebos and vaccination outcomes. That there are autism cases, very infrequent, but these models are perfect.
I have a question about the cluster 3 of the basolateral attention.
Yep.
You described them as distinguishing antecedents and consequences of actions.
Yep.
Two other words you could have used are causes and effects.
Yes, indeed.
Do you think that's what's being learned is causal, or is it just about the temporal precedence?
Oh, I know it's causal, but you have to be careful with the C word in behavioral neuroscience. I think it is. I mean, it is causal. That is, as it were, the modern take on instrumental learning. What I'm learning is the causal relationship between my actions and their outcomes. It's just that we can show that that's contingency specific. So as the contingency changes, so does the activity of the cells, so does the animal's behavior. But what we can't show at the moment is that it depends on something, say, like the value of the outcome, which you can do with rewards very easily, but with aversive events and punishes, it's a little bit more difficult. Yeah. So yeah, cause and effect.
So to rephrase it, that's a nod to sort of theoretical fights in associative learning, not because you have reservations about causing it.
No, exactly. Yeah. So antecedents and consequences are just less loaded terms in one of the fields that I work in. Yeah.
I was really curious about the idea that perhaps the problem is probabilistic learning. And I wonder whether, because you gave the example when the punishments are so rare, and then we've got all these people sticking to getting more punishment. So I wonder whether there is an issue there with temporal integration, because if life is short, then rare risks, pay off, But if you are able to integrate things over time and think, in the long run, this is not going to be a good outcome, then perhaps that's when you should be able to be more sensitive.
Yes.
So I wonder is there any other data that any way that we could look into that to see whether is there an issue of being able to think about the long run and integrating that probability.
Yeah, and I agree that's important, but there's also another, we talk about it in this paper, there's also another useful protective fact which is for most problem behaviours, the adverse consequences escalate over long periods of time. They start out very modest, thank goodness. I might, instead of running for an hour on a treadmill, go feed machines and pokies. I'll lose a little bit of money, but I'm not going to lose a lot. But across time, that behaviour could escalate, my losses could down, etc. That's certainly the case in substance use and other disorders, where the adverse consequences initially are really modest, but across time they escalate. The problem with escalating negative consequences is that escalation undermines our ability to learn about those consequences. And we've shown this in mice and rats, and you can show it in people, which is that, say, a punisher of particular intensity out here is there's path dependency in how that punisher changes your behaviour. If you're ramping up to that punisher, so you're living in a world where the adverse consequences escalate across months or years, doesn't matter. That punisher is far less effective at changing your behaviour than if you simply started with that higher level punisher from the outset. So yeah, I think there's a significant potential issue with integration, not just bridging the interval between, in this case, between actions and negative consequences, which can be delayed. But in our cases, they're not, and they show you that problem is present even when they're relatively immediate. But the other problem is that adverse consequences, fortunately or unfortunately, they escalate, and that in itself is a significant impediment to our ability to learn about those consequences. Yeah, good question.
Yeah. In that paradigm, the choices are always between something that has the potential to reward and punish, and then another option. And what's the contingency for reward with the other choice?
It's always matched as the same as both choices. So we'll do I think it's in the human stuff, it's normally 50%.
And so 50% of the time it provides a reward, 50% of the time
the probability of punishment changes across the experiment, sometimes 20%, sometimes 40%, sometimes 10%, but yeah. But the reward of both choices is the same, 50%, 50%.
But in the other choice, the not punishing choice, what's happening when they're not rewarded?
Nothing. So they're just partially reinforced. Yeah.
So it's a neutral outcome.
Yeah. Yeah.
That makes sense. So if I was a politician and I was interpreting this data, would it be fair to say that there is a group of people for whom compulsive decision making isn't going to be deterred by the magnitude of a punishment?
Yes.
So death penalties are probably not an effective way to prevent compulsive justice. And alternatively, it would support the creation of a police state, because the frequency of punishment should deter behaviour.
Yes, but this has always been known as a word, right? I mean, we've known forever that you know, it's the opposite to reward. Right? If you want a behaviour to persist, partially reinforce it. Right? So don't ever, like with kids, never just deliberately partially reinforce the behaviours that you want, and those behaviours will persist forever. If you want a behaviour to cease, then you need to be on top of it all the time. Yeah.
Right, we might call into the formal questions there. Basically, you're not really keep talking with our speakers. But yeah, please join me in thanking again our speakers, G. Yeah.